So I'm going to tell how we adapted Cas-CRISPR technology for malaria parasites. So a very brief introduction. Malaria is a very big problem, and health problem. So in developing countries, half of the world population is at risk of malaria, with more than 500,000 people dying of malaria, mainly children. So malaria is caused by a unicellular eukaryote. It's a protozoa called Plasmodium falciparum, and this protozoa shares his life cycle between the mosquito and also between the human host. So in the laboratory, we are able to reproduce this cycle. So, we can culture Plasmodium falciparum with human blood from donors, and we can reproduce this part of the cycle. In fact, it's the most important part because it's the part where all the symptoms of malaria appear. So I'm sorry, Hervé, I don't work in mosquito, so I'm not going to talk about the gene drive strategy in mosquito because we work really with the parasite that causes malaria. The mosquito only transmits the disease, but the parasite causes malaria. Okay? So as you can imagine, it's essential to be able to manipulate the genome of Plasmodium to study the parasite. And before CRISPR, the genome of Plasmodium has been very, very refractory to manipulation. So it was a really nightmare for the researcher, for us. So in a classical strategy in Plasmodium falciparum, before the Cas-CRISPR, we transfect the parasite with a plasmid where a drug-selected marker is flanked by homology regions of our gene of interest that we want to knock out. So then we had to wait for a spontaneous double-strand break to happen in the gene of interest. When this spontaneous double-strand break happens, then the cell needs to repair this double-strand break, and it will use the donor DNA that we provide in the plasmid. And with this process, the gene will be deleted or disrupted, and we have a knockout. So how we can speed up this process? How we can make it faster? We can produce ourselves the double-strand break, and like that, we can have an increase in the molecular recombination pathway. So for that, we have the CRISPR. The Cas-CRISPR allows us to produce that double-strand break that before we need to wait for the spontaneous to happen. So if we can produce double-strand break, we can speed up the process. So what do we need to adapt Cas-CRISPR for Plasmodium? So as we have been mentioning this morning, we need the Cas protein, but we need also the RNA. And the RNA, I don't know, it has been said, the RNA cannot be expressed by a Pol II polymerase. The RNA needs to be expressed by another polymerase because it needs not to have the cap and it needs not to have the poly A. This is the typical features of a messenger RNA from Pol II. So then we need to express the guide RNA from a Pol III promoter or from other promoter, but the Pol III is the more used one, as we have been commenting this morning, and the U6 is one of the more used ones. So we need the Cas protein, we need the guide RNA, and we need to provide the donor DNA that's going to be used as a template for the molecular recombination pathway. So we have been also commenting the two main pathways that we have to repair double-strand break. In Plasmodium, the non-homologous joining is very, very inefficient. So the main pathway to repair double-strand break is the molecular recombination. This can be great if we are talking about off-target activity, and we can discuss that later. This can be great to improve the efficiency of the system also, but it can be a problem if we want to do screenings. Okay, and we can discuss that later, but we don't have time now. So the strategy that we follow is a two-plasmid strategy. We have one plasmid that expresses the Cas9 protein and another plasmid that expresses the guide RNA. This other plasmid also carries the donor DNA that's going to be used as a template to repair the double-strand break. So we transfect the parasite with these two plasmids, and then these two elements will be expressed. The guide RNA will guide the Cas9 to the gene of interest, will produce a double-strand break. This double-strand break is lethal for the cell. It needs to be repaired. The cell will use the donor DNA that we provide as a template, and in this process, the gene of interest will be disrupted. So we were really, really happy and very excited because we moved from several months to several weeks to get a knockout. But we wanted to go further. It was great, but we could already do knockouts in the community. We wanted to do like surgery. We want to change just one nucleotide and not any other thing. We don't want to integrate any drug-selected marker. We wanted to do marker-free genome edition. So our laboratory is interested in the gene regulation, and then in the literature there was this publication where this protein, or one, is playing as a transcriptional factor in the regulation of a very important family of genes in this parasite. This is the virgin family. It's a virulence factor, and then they claim from in vitro experiment that the change from this leucine to alanine, it can abolish the binding of the ORG1 to the DNA. So we wanted to check if this is true, but in vivo, we want to create parasites that have only this change. So we follow this strategy. We have the one plasmid expressing the Cas9, the other plasmid expressing the guide RNA, and the donor DNA carry 2 mutations. So the Cas9 will provoke the double-strand break. The double-strand break is lethal. The cell needs to repair the double-strand break. It will use the donor DNA. And in donor DNA, we have the desired mutation, this change to the leucine to alanine, but we have an additional mutation, what we call the shield mutation. The shield mutation is just a mutation that doesn't change the amino acid sequence, but it changes the DNA sequence. So like that, and this mutation is exactly in the sequence of the guide RNA. So in the DNA, it's the sequence complementary to the guide RNA. So like that, the locus, one has been edited, now become resistant to the action of the Cas9, because Cas9 cannot recognize the locus any longer, because the locus carry this shield mutation that protects the locus to be recognized by Cas9. In this way, we can transfect the parasite with these 2 plasmids. We can keep the drug to maintain the plasmid in the parasite. So the parasite will express the 2 elements. This will happen, and only those parasites that repair the double-strand break with this donor DNA would be able to survive, because now the Cas9 cannot recognize the locus any longer. And now that we have the locus edited, we can remove the drug that maintains the plasmid, and then the cell will lose the plasmid, and now we have a strain that doesn't carry any drug selector marker and only have the changes that we wanted in the genome. So we check this by sequencing, as you can imagine, and then we have here the amino acid that we wanted to do, leucine to alanine, and then here we have changing CCA, CGA, so we change the sequence of the DNA, so the locus is not recognized by Cas-CRISPR any longer, but we don't change the amino acid. And it was done with 100% efficiency in this case. So another application that we did with the Cas-CRISPR was to study drug causality in Plasmodium. So this is a very, very health problem now. We have a very good drug that is artemisinin, that it was Nobel Prize recently, but the problem is that in Southeast Asia, resistance to this drug appeared in 2009. And this resistant parasite can be spread all over the world very quickly. That will be a huge problem. So in 2014, a group from Pasteur, they identified by association study some modification, some mutation in a gene that could be responsible for the resistance to artemisinin. But the real experiment was missing. What is the real experiment? The real experiment is I have sensitive parasites, then I change that amino acid, and then I check if they become resistant to artemisinin. So this, before Cas-CRISPR, was impossible to do, or was like 1.5 years of experiment. So what we did is we used the Cas-CRISPR to change just that amino acid, and then you can see here, so here is the mutation of the amino acid from cytosine to alanine, and here are the shield mutation. These mutations, as you can see, are very close, what Hervé was saying, in the seed sequence, very close to the very important nucleotides. So like that, this locus will not be longer recognized by Cas-CRISPR anymore. So we did it, and we create parasite only with that mutation, and then we check if they were resistant to artemisinin, and effectively, only that mutation make the parasite resistant to artemisinin. So we also mentioned the off-target activity in Plasmodium falciparum. We have edited several parasites, and then we did whole genome-wide sequencing to check if the Cas-CRISPR were doing other things elsewhere that we don't want to. And then we realized that in Plasmodium falciparum, virtually there is no any off-target activity. So we could not find any other mutation, only the one that were expected. And this can be explained because of the absence of non-homologous joining, and we can discuss that later. Also, we have been commenting the Cas9 application, so I'm not going to go through. So, David explained more about prokaryotes. So in eukaryotes, and also Hervé mentioned that we can fuse our dead Cas9 to another domain, any enzymatic activity that you want or whatever. So you can fuse to GFP, and then you will have a way to visualize loci in the genome by immunofluorescence, or you can also fuse to an activation domain or to a repression domain or to a epigenetic effector, or even you can target your dead Cas9 to a specific locus, and then you can immunoprecipitate that locus to identify proteins associated with that locus. That will be also very, very, very helpful in many cases. So also when I started my lab here in Montpellier,
So in our Leishmania lab, we try also to, in collaboration with Yvonne Sterke, also from MIVEGEC, we introduced the CRISPR-Cas also in Leishmania parasite, and these are the people involved and the funding. Thank you very much.